Thank you. I always forget that part.
I think we're good to go.
Okay, great.
Thank you very
So here are a bunch
much.
of Lego models of cars, right? So what can we learn about cars from popular Lego sets? Well, we see that spoilers are common, open roofs are common, bat motifs aren't unusual, right? But if we look at a regular parking lot, and here's one from exciting Cleveland, right? We find that there are very few Batmobiles in the world, right? Instead, there's many more minivans. Yet Lego doesn't make models of minivans, right? And why is this? It's because like what people want to buy is what's interesting. They focus on that. And that gets us to our talk today, the effects of taxon selection bias, studying what is cool isn't cool. And this is co-authored with Jeremy Bellew, who've been working with on this sort of area together for the last 20 years. So yeah, it's exciting. And so in biology, we often study sort of charismatic key clades, right? So orchids have amazing pollination behavior of biology, right? If anyone's seen any talks in this session, in any session on evolution, they've seen an anolis talk, right? Silver swords are really cool adaptive radiation. So we focus on these key groups. And history used to work this way too. So here we have a picture of Emperor Maximilian and Henry VIII, right? And so people often focus on these like popular celebrities, right? Um, but now historians take a broader view, right? And they might look at more focal examples, still missing more groups of people, but still better than it had been, right? Or you look at the whole frame and like how does this whole economy work? And we have to face the sort of same question in biology, right? So our mission in biology is to understand things like how life evolves through time, right? What affects speciation? How do genomes change? How do traits evolve? What leads to cooperation? And so these are general questions. We study them with particular taxa. And does that matter? Hmm. So one pattern that sort of made us think about this was this paper from Gingrich in 1983, looking at rates of change, the body size evolution rate on the y-axis versus time on the x-axis. And he found this sort of hollow curve. And on a log-log plot, it's a line, right? And so we see this in this, we see this in other models too. To play with this, we looked at a diversification model where we simulated a tree with a constant rate, picked random taxa from that tree, um, and then looked at reconstructed rates from that. So it's the same sort of hollow curve, even though we knew the truth here was everything was evolving at the same rate, right? So we mulled this for a few years, right? Other people went and got molecular data to look at this, morphological data to look at this, diversification data, fossil origination data, and found with the same pattern of these hollow curves where the lower left quadrant, the upper right quadrant are missing data, right? And why is this? And we do have some hypotheses about this, right? One is an artifact, right? So you're plotting a ratio against its denominator that's been known since 1893 to be problematic and lead to weird, spurious correlations, right? So what we do. Another is biased estimators, right? So maybe the tools we're using to measure things are themselves wrong. Another is an artifact of what we choose to study, right? So it could be the stuff in the upper right is just too fast, right? You don't measure morphological evolution between jellyfish and snakes. There's too much change. You don't until recently look at change over redwoods over 4 years because not much changes, right? So you sort of omit the things that are too much or too little, and you get this sort of medium zone in the middle, right? And of course, the fourth possible hypothesis is actually how biology works, right? Where everything is slower over longer intervals. The weird thing is that long intervals are made up of just small intervals that then are themselves fast. So it doesn't quite work, but it could be cool, and it could be cool if it's true. So one way to investigate this was to look at this ratio against this denominator artifact, right? And we took we have these rates and we have time, and so we can multiply rate times time to get amount of change. And we did a simple randomization test and said if we reassort rates with times, what do we get? And when we do that, we find these random things look very much like the original data. Okay, so it's almost indistinguishable. So this seems to be explaining a lot of the pattern. Now, this isn't complete, right? We actually made a full model called the HMB model, where we can separate this sort of noise effect that causes this hyperbolic pattern from a linear change in rate or a constant rate. And I'll give a poster on this at Evolution in Cleveland next month. Okay. Um, and actually, when you take out this noise effect, you actually still see there's some pattern here where that green arrow is. So come to Evolution if you care. So artifact one, plotting ratio against the denominator. Yep, seems like it could be an important thing. What about bias estimators? So Tanya Stowler did a great paper in 2013 looking at how different methods for looking at diversification rates calculate likelihood. And the scary thing is that different models, like what Ape uses and what Geiger uses, they use different equations that could lead to different optima. Okay, so that's one potential issue. Another issue is that likelihood is a great estimator, and Bayesian, its fancier wrapper, also a great estimator, but they can themselves be biased. So at large population size, at large sample sizes, they're great. At small sample sizes, they can have a bias. And we see this even with like the normal distribution, looking at standard deviation, right? And even that with as many as 50 points, you can still get a biased estimate of standard deviation by using the likelihood estimator. So we have an unbiased estimator for standard deviation. So what about in phylogenetics? So we often think that we have big trees now, but there's a limit, right? There's only so many cetacean species we have, right? And also there's all these methods like clads or Missy or Bama or Medusa that split trees into small components that have their own rates, right? So this has a whole tree has 87 species. This one clade has 6 species, right? And so you imagine that there could be a bias there. And so we published a paper this year where we actually did the math and had a computer help us with the math to figure out what these biases are. And we actually found corrections for the biases. So now if you want to have an unbiased estimator, you have one, which is great for when you're comparing different clades, right? Because otherwise, a smaller clade will have a different rate than a bigger one, even though they have the same true underlying rate because of this bias. However, this bias we see doesn't match the empirical bias. So it's important to correct for, but it's in a different direction than what we expected, right? So we have a correction, but the bias is in the wrong direction to explain this. And then what I think matters for top number 3, what we choose to study, right? And so there are various ways that we choose what to study and their effects, right? So one bias is survivorship, right? So for modern biologists, we don't study extinct things very often. And so, that creates a bias, right? So like we're trying to estimate, death rate for humans, right? Everything in this meeting is alive, right? And so death rate for humans is 0. Cool. And we know it's not true. And there's corrections for that in various diversification models, but still something you can consider. Another one is variable clades, right? If I want to study gain or loss of woodiness, I don't do it in oaks because they're all woody. I don't do it in whales because none of them are woody, right? So I try to optimize for the clades that have enough variation, and that creates a bias. And Lewis came up with a correction for this in 2001, right? The MKV model. And that's a correction for, excluding clades that have no variation, but it doesn't really deal with if you exclude clades that have only little variation or too much variation, right? So we have some corrections, not a lot. Name-based, right? People focus on orchid or orchidaceae, right? Not a few nodes, rootwood or tipwood on the tree, right? And so maybe there's some weirdness about how we're naming clades, right? Maybe they have a long branch and a lot of apomorphies on that branch that could lead to a bias in what we're looking at in life. Practicality, right? You don't have your grad student go work on generation time in Sequoia because they'll never finish. Charismatic clades, right? So as shown by tattoo incidences, right? So like people want to study certain clades and everyone wants to get on those certain clades, which is great, but that means the other clades are unloved and don't get as much study, right? That can be a sort of a pile-on effect. And finally, an organism first thing, right? Where people say, I want to study blah. Like for me, for grad school, I want to study ants. And so I chose ants and I chose a good group to answer good questions within ants, right? But I was an ant person first, a question person later. And that can affect the sort of questions you're asking, which clades we're asking them about. So we would try to test some of this, right? So the name-based thing, one thing we tried was took a campaign tree made by Jeremy and colleagues, and then simulated evolution of morphology on a constant rate under that tree, and then compared a random clade on that tree with a name clade of the same size, right? So we some genus would find a similar random clade that had the same size of that genus and compare their results. And it could be that they had no difference in rate reconstruction. It could be the name group was much slower or the name group was much faster. And so each line shows one comparison of a pair. And here's a bunch of lines across this trait. And to my surprise, we found no net trend. So like, even though I was worried about this naming thing causing a bias, it didn't cause a bias. What it did cause was more variance. And so the name clades had more variance in the rates than the random clades, even though they had the same true underlying rate, but there's no net bias in that. We also looked at sort of this other thing of like which clades are studied based on charisma or some other practical reasons. And so to study this, we got every information, every paper that cites clads, BISI, HISI, or BAM. So we want to get basically diversification only models, so not like Rev Bayes that does a lot of different stuff. Filter for ones that are in plant journals. Pull all the names of all the scientific groups in the abstract and titles and keywords. Select those are vascular plants. Make sure it's only empirical plant papers. So check them all manually. Use a plant tree. Pick 5,000 random nodes in that tree, and then pick nodes that match the clades that are used in the empirical papers. right? And they finally used Magyar and Sanderson to estimate net diversification rate. And so here we see the results of that. So red is the actual empirical ones that people used. Gray are all the empirical ones that people, just do that were chose at random. And you can see that papers study these clades, which have a large number of taxa and a large age, but most of life is over here in these clades. Okay, so there's definitely a bias in what people are choosing to study. We reconstruct the rates too. So the random ones and the stem lengths, and so the random ones have much shorter stem lengths, the branches leading to the clade origin, than the studied ones. And the seed plants look like they have a faster diversification rate, but on average, most of them are much slower, right? And so here we see this bias from what we choose to study that doesn't represent plants as a whole. Okay, one of the only ones who have worked on this. So Meyer and colleagues studied this looking at mating systems in plants. And they found that the plants people actually study tend to be biased towards having a higher rate of hermaphroditism than plants in general, right? So you may be misled about what plants do based on what people study rather than what's out there. Okay. Um, let's look at upgrades studied by charisma or other reasons. So similar sort of thing. We got 73,000 papers. We pulled all the names of taxa from those. We got the family, air quotes, from them. Um, we got all the species per family. We got all the papers containing the family or any of the taxa, genera, or species within it, right? We got number of species in GBIF, number of species in GenBank, and then plotted it all, right? And so here we see a few cool things. One is that there is a correlation between number of species in a clade and number of papers about the clade, right? But it's not linear, it goes up faster than linear. So the bigger thing is, more people study it. Also, for a given sample, for the number of species, there's variation in how many papers are on it, right? So just sort of zooming in on one of those. I studied ants for my PhD. I love ants. Ants are great. But Crambidae, so grass moths, are about as diverse, but they have maybe 1/10 the papers. Fewer species in GenBank, fewer GBIF records, even fewer phylopics, right? And so same clade clot size, similar clade age actually, and yet one's been studied a lot more. So we're trying to understand how life works. It's biased towards what ants do rather than what grass moths do. And so artifact three, there might be a thing there, right? This can't rule that out yet, which makes it hard to then still look at biology, right? And see like what's true about the biology if we haven't dealt with all these other biases first. How are we going to solve this? One, we're going to get NSF funding. So think good thoughts, but you know how things go. We'll do it anyway, but it's nice to have funding to support postdocs and mentor and things like that. Um, Run analyses of many clades across random clades and across named clades to see how those differ. Look at well-studied and poorly studied clades, right? So are well-studied clades for certain things faster rates than others, right? Are they more, faster diversification or something else? And sort of see what these patterns are so we can get back from that to figure out how life actually works. Discover what's common across the tree of life, and then also sort of a talent search for new clades, right? So find ones that are good for particular questions. Um, yeah, and so that's my talk. And so thank you NSF for lots of money over the years. Thanks for Nimbus for support and specifically for support for Jeremy. iPlant for support for a lot of these things. And like this grass moth saying, nod vigorously if you think these understudied clades need more work. Thank you.
Great. Thank you very much, Brian. Are there any questions? Okay. I think we have still some time. If you have any questions, feel free to raise your hand and open your mic and camera, or you can put any questions you have in the Q&A.
I have a questions.
Yeah.
With the correction factor that you show with this for the speciation and extinction rates, is that for for the constant rate, but and that can you do it for with the trade dependent, for example, rates as well?
You would do it for, if you have a trait dependent one, like if it's state 0, you correct the state 0 speciation rate. State 1, you correct the state 1 speciation rate. We haven't done a sort of quasi correction yet. Or presumably work the same way.
Okay.
Yeah.
Thank you.
Great, thank you. Albert, if you want to share your screen, and yes.
Okay.
Thank you again, Brian. Our next speaker is Albert Soewongsono. Sorry.
It's
Don't
okay.
pronounce that. So we're good to go.
All right. So, okay. Thank you. Hi, everyone. My name is Albert. I'm currently a postdoc in Michael Landis lab at Washington University in St. Louis. Today, my talk is about distinct evolutionary processes, identical T patterns, and their mapping in SSE models. So as we know that our planet has a lot of species diversity, and sometimes you can group the species according to a certain character, their states. Maybe they are life young or they are egg bearing. And when you look at their phylogenetic tree, you often see something that looks like this thing, like an imbalance of in terms of the number of species in different states. And so the question is that we want to answer is that why we see more, for example, blue species than species in red. So one way to answer that question is to have a model that links species diversifications with trait evolutions under one framework. So the state-dependent speciation and extinction model class does exactly does that. And then over the years, you have these different variants, such as like VC that models a single character with two states. You also have GeoSSE that models biogeographical process. And then you also have PC, of course, that accounts for hidden process. So what you do is basically you have your information from your phylogenetic patterns that can be used to inform the underlying process. So here we think like a backward in time where we treat model as inference. So you see from your phylogenetic tree, maybe you have the character data at the present, the thing that you can observe, you can gather, and then you have your phylogenetic information such as their topology and then their branch length. And then so you want to feed the data into a generic model like a BirdNet model, and you want to use your favorite inference tools such as Bayesian phylogenetics or maximum likelihood to get something such as evolutionary scenario. So these are the evolutionary scenario that best support your data. Maybe you get something that looks like this, where you have a positive net diversification in one of the states and the higher transition going from the one state to the other states or the other way around. So what I'm gonna talk today is that what if you do not have that information? What happens if you do not have a tree? So what happens if we only know about the distribution of species in different states? So can we say anything about the process just by knowing that information basically? So here we think of a model as the data generating process instead of as an inference. So we think forward in time. So if you have a like evolutionary scenario that looks like this, if we simulate trees under this setting, maybe we get this type of realizations where you have an equal split in terms of the number of species in A and B. But maybe there's a competing scenario that looks different than the first scenario, the scenario one, and then the competing scenario also leads to the same realizations. So as a general pictures, maybe what we actually have is that something that looks like this. So you have these three different classes in terms of how you can define the relationships of these splits between one state and the other state. Maybe you have equal splits. You have one state with more species or one state with fewer species. And in each of these classes, you have these different scenarios, these alternating scenarios. They all correspond to the same amount of splits in terms of the number of species in both states. So in this talk, I'm going to show you that these alternate scenarios do exist, and I'm going to use BC as a case example. And then each of these scenarios is governed by a unique rule set. So I'm going to explain what a rule set is next. So basically, what you get from your inference is something like this that looks like the one on the left, where you have a higher net diversification in A, lower net diversification in B. But you can also basically treat them as a rule set. So rule set here is just a functional relationship between model parameters. It consists of the set of equalities and set of inequalities. So you can draw these from this equation infinitely many solutions in terms of your speciation rates, your transition rate, and then they all will correspond to this exact split in terms of the number of species being in state A and in state B. And so in general, you have basically these bijections between rule set and also evolution scenarios. So in the class where you see more species in B than in A, you have 7 of them. So for example, the rule set 1A here will have a property where you have a positive net diversification in A and lower net diversification in B, with a higher transition going from A to B, and then this rule set 2A will have a different property than the rule set that is defined by scenario 1A, for example. So in total, you have 7 of them for this class, and then they all correspond to the exact number of splits in A and in B. And then if you look at the symmetry, if you're in the class where you see fewer species in B than in A, then you have exactly 7 of them as well. And if you are in the class where you see equal splits of species in B and in A, you have 3 of them. So in total, in the BC case, you'll have 17 different scenarios, which means you have a 17 unique rule sets that define them. So up to these points, basically, I'm saying that the multiple scenarios can produce identical stationary tip frequencies, and then there are infinitely many parameter combinations that can be drawn from these scenarios. So the next thing is basically how different these scenarios are in terms of generating valid trees, because of course, we want to generate non-extinct trees. This will be useful when you define your priors, for example, when you want to do inference. So in this case, we will draw parameters uniformly from like a hypercube. So in this case, this will be 6-dimensional hypercube because this is a VC example. So you have 6 parameters. And then so we will look at what the probability that this scenario in terms of generating valid trees. So this is a simulation study. So you can basically also group these different scenarios into these 4 different things. So it's like so you have 2 scenarios that correspond to sourcing dynamics. You have 2 scenarios that correspond to source, and then 2 scenarios that correspond to the sinking dynamic. So a state here is a source if the speciation rate in that state is higher than the extinction rate, and it's a sink state if the speciation rate is lower than your extinction rates. So this seems to be intuitively true that if you have a source of dynamics, then you are less likely to get a extinct tree because things will just exponentially grow over time. So in the next plot, is something that is different. So if you assuming you already have your set of valid trees, how do we assign the probability for these different scenarios to these valid trees? So now you see something that is a bit different than what the other plot is showing, because now it is still true that the scenario that corresponds to the source dynamics, that is scenario 3A, is being associated the most to the set of valid trees that you have. But in this case, something happens here because now you see more scenario 2A being associated to valid tree compared to scenario 4A, despite scenario 2A is the one with the property where the source and sink dynamic. So how do these 2 scenarios differ? So in scenario 4A and scenario 2A, they all belong to the same class where you have more species in B than species in A. However, in scenario 4A, the directionality of the transition rate is in the opposite. So you have more species going from B to A rather than the other way around. While in scenario 2A, these rule sets that correspond to this scenario do not have a restrictions in terms of their transitions relationships. So I can go more on this in Q&A if people are interested. So here, basically, I'm showing that each scenarios occupies parameter regions of unequal volumes in terms of their capability of producing valid non-extinct trees. And then also, I'm going to show you the inference result next, and then I'm going to claim now that not all scenarios are equally preferable to explain trees with only extant species. So when you look at these across literature papers that they use BC as an analysis, you see that all of them basically prefer this either scenario 3 or scenario 4. So these 2 scenarios are the ones with the property where you have 2 states, both states being the source states. So you have a higher rates in speciations compared to the extinction rates. We also do our empirical analysis using the squametes, families, and intra-orders. So one thing that is common here is that you can see that these scenario 3A and then 4A, the one with the source dynamics, are also the one that is mostly inferred. However, you also have some instances where the 2 scenarios that correspond to the source and sink dynamics are chosen as well. When we do the inference on the simulated data set where we know the true generating parameters, we see something that is also rather different than what you see using the empirical tree. Because now here you see that scenario 2A, the one that's with the property where one of the states being a sink state, is actually more more frequently chosen compared to this another scenario, which is scenario 4A here, which is another source and source scenario. So why? It has something to do with the fact that we are using extent-only trees and also these different likelihood conditions that people use when you do inference. So I'm happy to go through in the Q&A. Yes. So yeah. So in this case, when we look at these inference based on the empirical data set and an inference based on the simulated data sets, you can see here on the simulated data set, the number of instances where you get the scenario 4A is far less than one compared to the one where you use empirical trees on the right. So to summarize all these different figures, when we go from the simulations to the inference, basically the relationship that you see when you do simulations and then the pattern that you see when you do inference, they're not always one-to-one. When you do simulations and then in terms of getting the proportion of how easy each of these scenario in terms of generating valid trees, the one with the source dynamics are intuitively kind of plausible to generate more valid trees because you have a higher speciation rate than the extinction rates. But when you look at these, like if you already have a set of valid trees, not all the source scenarios, such as scenario 4A, is being associated more to these valid trees because some of these valid trees are actually associated to the other dynamics where you have a sync state in one of the states, such as like in scenario 2 here. And then when you look at the patterns when you do an inference, it's also different whether based on whether you are using empirical trees or you are using inference based on the simulated data set. So just to as a conclusion here, basically we have this theoretical framework that can allow you to link between the data generating process, which is the patterns that you see, and also the actual process that generates these patterns, so your model parameters. So here this is using SSE models, and then we use BC example as the proof of concepts, but you can generalize this in other more complex SSE models, such as, for example, in GeoSSE or Classy, for example. And then also these different simulation studies, when we simulate trees under different scenarios, this can be used as a guide when you do your inference, when you want to basically choose your priors on your parameters if you do a Bayesian phylogenetics. And so you want to basically choose your priors that are consistent with your observed data, which is the data that you see at the tips. And that's all for me. If you have questions, please let me know. Thank you.
Thank you very much, Albert. We still have 2 minutes for questions. If there are any questions, feel free to open your mics. Okay, I don't think there are any questions in the chat. Albert, very quickly, I do have a question. How do you, maybe I've missed that, I'm sorry if I did, but how do you separate the general bias you identify in the simulations from genuine biological support for the diversification scenario?
Sorry, can you say again?
Yeah, how do you separate the general bias that you identify in the simulations from the genuine biological support?
Oh, well, so the for the inference, we also do it on the simulated data set, which means that we know the true J parameters. So we can basically know that the scenarios that correspond to the true J process is the same with the inferred scenarios that you get from your inferred parameters when you do an inference. And so you can be sure that these are the correct scenarios. And then we also do a confusion matrix to see the false positive, false negative under SSE models. And we don't really see a false positive here. So it's not an issue. But in terms of like empirical trees, well, you don't know the true J parameters. So maybe this discrepancy that you see here between the scenarios that are being chosen by preferred by the estimated data set, empirical data set compared to the estimated data set, maybe due to the biases, for example. Like
Okay.
here, you can see that empirical here, you tend to you see more scenarios for A being chosen compared to like on the estimated data set, you see more scenario to A. But you can be sure this is true because we know
We
the
have
true
to move
generate.
on. So sorry.
Yeah, sorry. Yeah. Thank you.
But